Okay, I'm going to sit down for this one in hopes that you can see this and me at the same time and be able to see what the compartments look like. Now, in this particular lab, we're going to be looking at this multi-test system, which allows doctors to determine the bacterial cause of intestinal symptoms. So, for example, if a patient comes in and they have vomiting, diarrhea, abdominal cramps, pain, gas, indigestion, all that kind of stuff, that could be indicative of a viral infection, a bacterial infection, all different things. So a doctor would want to determine which type of infection the patient has in order to treat him with the correct antibiotics. And remember that enterics are bacteria that are common in the human digestive tract, but some of them can become opportunistic pathogens. E. coli, Enterobacter, Pseudomonas, all these different ones can be pathogenic if you get them in the wrong doses. Clostridium, Clostridium is not a normal microbiota. Or also you would want to find out if maybe the patient had something like Salmonella, which is not a common microbiota. So you can't just go by symptoms to determine what a patient has. Symptoms may be the same for two different microbes. So this system right here was designed to test very rapidly, meaning within 12 to 24 hours, the exact microbe that a person has and to treat it with the correct antibiotics. Because remember, we want to try to avoid the use of broad spectrum antibiotics because of the problem with resistance. And figuring it out within 24 hours what the patient has is a better route than just giving them some antibiotics in hopes that it treats the infection. The other thing that that this is it's easier to use because it has multiple tests in it. And if you recall, we've gone through this whole process of using staining techniques to identify a bacteria, using biochemical tests to identify a bacteria, and we went step by step by step. We did fermentation tests, we did protein catabolism, we did differential media. This right here, this one tube, allows the doctor to look at multiple reactions at one time. So we have fermentation reactions, we have the urea test, we have a citrate test that we haven't done yet. But these right here, you can actually determine the exact cause of the symptoms with this one testing system. Another test that can be used is the IMV-IC test, and that's actually a series of tubes that would be done. And it gives you kind of the same results, but it's not quite as specific. With the IMV-IC, you can get it narrowed down, but there's multiple bacteria that can still show the same results. And also they can be variable. This is much more specific. So what you're going to do with this one right here is, hopefully you can see, but we have you can see the colors. And so here we have the differential, the different media, the differential media, like the fermentation tests. And then we have a protein test and some other ones down here. With these particular tests, you're going to run your bacteria through this entire thing, and it will change color or not change color in each little compartment depending on the actual species of bacteria. The other thing that I want you to notice is these first three compartments, they have this white stuff in there. That's actually wax. And remember that some bacteria, when they ferment sugars, they produce gas like carbon dioxide. And what will happen is the wax will actually separate from the media if the bacteria produces gas, okay? At the very least, a microbe that is an enteric is going to ferment glucose and probably produce gas. So your first compartment should always change color. If it doesn't change color, then it's not really an enteric bacteria. The enterics are all gram-negative. When we did chapter 11 with the dichotomous key, we talked about how the enterics all fit into that family that has flagella, and they're gram-negative. They're mostly rods, and that one of the other characteristics is they do ferment glucose. So at the very least, you'll see a color change in this first compartment, unless you're giving a bacteria that is not an enteric. Okay, so for this lab, for each group, you're not going to have On the plates that you have, it's not going to have the name of the bacteria. It's going to have a number or a letter. So it's going to be an unknown. It's going to be one of the bacteria in the lab, unless they throw in some sort of unknown that we don't know about. But it should be either E. coli, Salmonella, that Typhimurium we did earlier, Klebsiella pneumoniae, Enterobacter aerogenes, the Shigella we don't have, or Proteus vulgaris. So the Shigella is the one that we don't have. We've tried to use it in other labs, and that was one bacteria we were not able to obtain. So each group will get one plate of unknown and one Enterotube. Unless we have enough, we might split it up for every 2 people. So what you're going to do with this particular tube here is you're going to take the colony, and so this will have bacteria on it that the lab tech would have done that streak plate method where they made isolated colonies. So what you're going to want to do is grab an isolated colony. You don't want from the smear, you want to get one of those little ones out by itself. Now with the lab manual, it tells you some things like removing tape. Well, ours doesn't have tape on it, so we have basically the same tube, but the procedure might be a tad bit different than what we have in the lab manual. So what you have here, and of course I have my imaginary goggles on and I have my imaginary gloves on. At this point we don't need a flame. We're going to need that in the second lab. So for this, what you're going to do is, if you look, there's 2 caps on this. There's a whitish clear cap which has a pointed, the needle is pointed on this side. I guess it's a needle. And on the other side, in the blue cap, there's what looks like a loop. Okay? So what you're going to do is actually remove the caps, and you can set them down. And you're not going to flame anything. This thing is already sterile. And you're going to actually, this side that's pointed is actually the part that you pick the bacteria up with. And so you're not going to flame it. You're just going to go and you're going to pick up your colony. And like I said, only grab one isolated colony. It is kind of pointed, so try not to gouge the auger. And then you're going to turn it and pull it through as you're turning. Okay, and you're going to go all the way to the end, but don't pull it all the way out. Okay, where's the end of it? Remember to turn as you pull. And then you go all the way to the end, and then you go back through the exact same way. So you've basically gone through all the compartments twice. Okay, now we can actually put this cap back on. Okay, and you don't have to do it real tight, just close it. Now what you want to do is pull it, and I know you can't see this in the video, but right here, so here's the loop side of it, there's a little tiny notch, and I can feel it with my finger now. You're going to put it to where that notch is even with the end of the tube. And you're going to, away from you, you're going to snap it, and it will literally break off. Okay? Now we can put the cap back on this side. Okay, once again, I'm not working with any bacteria. It's all imaginary. Okay? Now, this is the part where it's different from the lab manual. The lab manual says remove the blue tape. Well, we don't have any blue tape on ours, but the goal of this is with this part right here that you've just broken off. On the compartments, starting with citrate, all the way down to adonitol. So these first, not these last 4 compartments here, so starting with this green one down to this red one, okay? Notice that there are little notches right here, okay? And what you want to do is take that part that you had just broken off, and you're going to pierce a hole in that notch, okay, on each one. And essentially what you're doing is exposing these parts to oxygen. Because remember, like with the fermentation test and the urea test, we didn't tighten the lids all the way because we want them to get a little bit of oxygen. So what I've done is I've pierced the side from the red compartment here all the way to the green one here. Okay? Now you wouldn't be doing all this because you'd have bacteria and you'd be moving a little bit quicker than me. Do not pierce the part that has the plastic on the flat side. okay? So when you go to put it in the incubator, you're gonna put it down here. The compartments that you're gonna pierce are on the side of it, not on the bottom, okay? You want this to remain the way it is. So you're gonna take this, because it may have bacteria on it, and you're gonna throw this in a waste beaker, which is imaginary right here, okay? So then you're gonna take this whole thing and you're gonna label it with your name and your group over here, and you're gonna put it in the incubator that flat side down, okay? So when you come back the next time, you're gonna read your results. Now at this point you will have a flame. It's imaginary, see it's right here, and I'm wearing my goggles and my gloves. So what you're gonna do is you're gonna read your tests. So what you'll need for that is a couple of things. One of them is I'll have one of these, and I don't know how many I have, but I'll find as many as we can. This right here comes from the manufacturer, and it shows the uninoculated tube and then what the results can mean, okay? Now we are only doing the Enterotube. We are not doing the API one. We're only doing this one. In the lab manual, it has you do both of them and compare them, but we only have the one, okay? So some of the reagents you won't use because it's for the other test. So what it wants you to do is read all of the tests except for the indol and the VP. Those are more confirmatory tests. And so the indol is right here, H2S and indol, and the VP is right here. You may or may not need to do those depending on if you come out with a code without them. So what you're gonna do is based on this right here, and you also have a picture in your lab manual that shows the tube and what some of the changes can look like. It doesn't show all of them, but you can see some differences. For example, you can see some of these compartments that were yellow to begin with can turn purple, okay? Or this first one that was red to begin with will turn yellow. And so to read the results, the actual results are not in your lab manual. They're going to be in this little guy here. So I'll find as many of these as I can. So it's upside down. Did I have it upside down the whole time? I apologize. So this is the right side to read it. Now remember, you have an unknown. You don't know what it is. It's one of those that are on the list, and maybe if another one is thrown in. So in the uninoculated, you will notice that the first compartment is red and the last compartment is green. So after your 24 hours, you should see some color change. So positive results for the first one is red to yellow. And if you have gas, you'll see that separation. And literally the wax will move away from the media. You can see the second two will turn purple. And then on this fourth one, do you notice the black? That's a precipitate. So if it forms hydrogen sulfide, you'll see that black precipitate, okay? The indol part, you're gonna wait until later. And then you can see the next four will turn yellow. If you do the VP, you'll have a red color change. And then this one here, unfortunately, has both the dulcitol and the phenylalanine all in one compartment. So if you get a positive for the phenylalanine, the green will turn to brown, and you won't be able to tell if it turned yellow, okay? So if it turns yellow, that means dulcitol positive. If it turns brown, that's phenylalanine. So they read both of them with that one compartment, okay? Like I said, if you get the brown, you won't be able to determine yellow. And then for the urea, it shows pink here. And remember, that was that hot fuchsia pink. So if it goes from this orange to hot fuchsia, and then the citrate we haven't done yet, but if it uses citrate as its source of carbon, this will turn blue, okay? So you might have them all change, you may have some change and not others, and that means something. So the next thing you're gonna do is you're gonna read, when you read your results, you wanna look in your lab manual, and you're gonna fill out this little guy right here, okay? And if you have a positive number, so pretend that this turned yellow and it had gas. So you'd actually circle the number below glucose, the 2, and the gas for 1. And then notice also that there's a bracket. So you'd add those two together and you'd get a 3. So like I said, your first number will be either a 2 or a 3. It should be. And then you keep going along. So say I had lysine change, but I didn't have ornithine, but I had H2S. So I'd circle the 4 and then the 1. I'd add them together and my next number would be 5. On the next one, let's say we had negative for indole, positive for lactose, then that would be a 1. And you just keep doing that until you actually come up with a 5-number code. You don't add these numbers up, you actually write the code right here. So it'll be a 5-digit code. And then with your 5-digit code, you come up to me and I have this booklet, and I couldn't find it today. Hopefully it will be here. I'll have this booklet and I will look your code up in the booklet and it will tell me exactly what bacteria you have. Now, if the code doesn't match any of them, I'll have you bring your little guy up to me and we'll interpret the results together. Sometimes it doesn't interpret the same for everybody. Maybe this goes more pink than yellow. What does that mean? So we'll interpret it and I'll ask you how you got your number. And we'll go, well, is that really gas or is it not? Okay, and we'll determine all of that. And then we'll look to see if your code really matched. If you need to do a confirmation, then we'll want to do the indole and the VP test. Now in the lab manual, it actually tells you to use a syringe to inject your stuff into there. But we don't have the syringe. What we're going to do is for the indole test, which is this 4th compartment here, you're actually going to take a loop and you're going to flame the loop until it gets nice and red, and you're going to pierce the plastic, putting a hole in the plastic. And then you'll take the reagent, which is usually called Kovacs reagent, but ours actually says indole. And this is one of those little ampules that you have to break them, and then you squeeze it and you just drop a drop in there. Okay, and you should see a result pretty quickly with that one. I'm trying to remember. Look to see in your manual if it says to wait a certain amount of time. Okay, the other one that you can do is the VP test. So if necessary, you'll do the same thing with the VP compartment, which is the 4th to last. And once again, you'll use the flame and you'll burn a hole in it. And then the VP reagent B, so is what you will use. And same thing, you'll break the ampule and you'll squeeze it and drop a drop in there. This you actually have to wait 20 minutes for a result. So hopefully, if you have to do it, you have other things that you can do during that 20-minute time. Okay, that will come up with a red color if you need to do that one. And then maybe this one came up positive and that didn't, and then we'd add a number, we'd change the number. Okay, so it works pretty well. Sometimes we have 100% success and other times we don't. Sometimes there's just errors here and there. So with this, like I said, you'll have the exact species from the code book that I've given you. And this is really a fun test for a couple of reasons, because you get, well, the color changes are pretty, but it's like actual real tests that would be done in a lab, and you get multiple results at one time, and it's usually pretty accurate. And when you see the book that I have, there's multiple codes that will work for one bacteria, and it's pretty specific. So it's a nice test because you get those specific results. And so that's the Enterotube, the multi-test system.